I think there are four aspects that contribute to what makes us human, and you see them here. But to be honest, I frankly admit that I picked this specific aspect not only because they contribute to our humanity, because that in one way or another, they relate to brain projects that are going on in my lab, and I would like to tell you about this brain project and how they relate to this aspect. So let's start with language. We had a nice session about it earlier. This is, of course, a very important and unique ability of the human species, and it's worth studying by itself. But as a brain scientist, I also want to consider it as a tool, a very, very powerful tool. I like to think about language as sort of an inbuilt microscope that we have in our head that converts activity of neurons and tell us about their meanings. So by asking the individuals what goes on in their mind, we can get insight into the meaning of their activity patterns, and I hope I'll illustrate this in the project that I'll be describing. So in addition to the tool of language, where we simply ask the individuals what goes on in their mind as they perform the experiment, we also record the activity, neuronal activity, and in my talk today I'll focus exclusively on one method, which is intracranial recording. This is an invasive method done for clinical purposes in epileptic patients, where the neurosurgeon is placing many, many contacts on the surface or sometimes even inside the brain, and you record other activity of groups of neurons or sometimes even activity of individual neurons using this method. The implantation is chronic for several days, and in between clinical sessions, the patients participate in cognitive experiments. Another introduction that I would like to do is about the visual system, that's the framework, and this is the system I'll be talking about. So I'll just give a very, very brief introductory overview. Simplistically thinking, you can think about the visual system as a ladder, hierarchical ladder, where the first area V1, is the lower rung on the ladder, where neurons are very simple receptive properties that are tuned to simple local elements in the pictures that is presented to the individual, like lines of different orientation. As you climb through this hierarchy of areas, the information gets integrated into more and more complex and gestalt-like patterns until you end up at the end of the hierarchy, that contains areas that are already selective to entire categories of images. For example, a very prominent area is a face area, where neurons are selective to faces and no other category. And nearby to it, there is a place area where neurons will be selective to place images and not to faces. And from here, the information transports to the hippocampal formation for memory processing. So this will be the system that I'll be considering in my presentation. Let's start with free behavior. And I would like to define free behavior very, very generally as the generation of a cognitive event in your mind in the absence of a specific external cue. And the question is, if you think about free behavior, you realize that it's not ever completely anarchic and free. Even when you are doing something completely spontaneously, it is always limited. And just think as an example of the phenomena when I'm asking you, name all the animals that you can think as fast as you can, and you'll easily come with a horse, a lion, a mosquito, whatever. But you'll notice that you rarely, if ever, will say tomato. So even though you're spontaneously coming up with many, many different names of animals, your brain knows how to delimit the spontaneous free behavior within a boundary and do not go beyond this boundary. We do it very easily and very flexibly. And the question is, what is the brain mechanism that is involved with this delineation of free behavior? And this question interests Itzik Norman in our lab, who conducted the following experiment to explore it. The experiment is fairly simple. It's actually following previous experiment we did with Agar Sagiv and Yitzhak Fried. In the first stage of the experiment, the patients are asked to view images. I bring you here examples of these images. These are vivid images of faces and places. In the actual experiment, they watch them one by one, inspect them several times so they can memorize them well, images of faces and places. So this is the viewing session when they actually see the pictures. After that, we move to the second stage. The eyes of the patients are covered, so they cannot see anything, and they are asked to, first of all, and this is where the language instruments comes in, they are asked to report into a microphone whatever comes to their mind. So we have an ongoing sort of commentary by the patients of all the cognitive events that happen in the mind, and we ask them to recall the images that they saw a moment before, but critically for the experiment, they are asked to recall the images in blocks. So they're asked to recall the faces in a separate block from the places. First recall all the faces, then the places, then the faces, and so forth. So we separate the target of recall into different categories, faces versus places. So what Itzik did next was to look in those cortical areas that I mentioned that are selected to the specific categories. So for example, you see here a response from an electrode in this face selective region, and you see that it responds to the images of faces. This is for the viewing period, when the patient actually saw the pictures, and you see it responds very, very strongly to faces and almost none to places. And in another electrode that was sensitive more to places, you see the inverse. Green is places, and red are faces. So these regions are selective during the viewing condition to faces or places. The critical question is what happens when the patient is trying to recall places or faces. And here the result was very striking. The moment the patient was instructed start recalling, we found a baseline shift, a general increase in a sort of the DC excitability of the relevant cortical representation. So when the patient was asked to recall faces, throughout the recall period, this is the entire recall period, there was a general increase in excitability in the face selective areas, and when he was asked to recall places, there was a suppression of this activity. And the inverse happened in the place selective areas. You can imagine, metaphorically, it's as if the brain is warming up the representation that is responsible for the recall and cooling down the representation that are irrelevant, and by this it is allowing a better or faster recall in the targeted category and preventing the spillover into the wrong category. But what about the actual events of recall? What is driving them? Here, a very attractive candidate is a phenomenon that was discovered several years ago, and it is very powerful, and these are high power synchronous bursts in the hippocampus, which are called slow wave ripples. They were described by Yuri Buzaki as the most synchronous event in the brain. Itzik managed to record them and find them also in the human hippocampus in those patients. And the question is whether these ripples, this burst of synchronous activity, are actually related to the recall of the images that the patient saw. So first of all, during viewing, what Itzik found was that ripples actually have content. There are electrodes that are recording ripples showed preference for certain number of images that we call the preferred images, and showed very weak ripple activity for the non-preferred items. These do not have to be faces or places, but a subset of the images activated ripples. They differ between different electrode recordings, but generally every recording had preferred ripples and non-preferred ripples. This is during viewing, when the patients were viewing the images, so you have preferred items activating ripples during viewing and non-preferred items not activating. And now the question is what happened during recall. Remember that we have a commentary. The patient is telling us what happens in their mind, so we can actually understand the cognitive meaning of these events of the ripples. And when you look, and what Itzik did here was simply separate the ripple appearance triggering related to the onset of the recall to the preferred items, the ones that were activating during the viewing, and the non-preferred items. And you see the result here, and before the preferred items were recalled, there was a gradual buildup of ripple activity starting 2 seconds before the actual recall event, anticipating or maybe perhaps even driving the appearance of the recalled item. I remind you, this is in complete absence of any external stimulus. For the non-preferred items, there was almost no increase. So we are now able, using language, to say that these ripples are actually associated with the preferred items that the patient is recalling. If you look now in the cortex, not in the hippocampus, Itzik found that there is actually a dialogue between the cortex and the hippocampus centered around the preferred ripples. So if you record in cortical site that had preference for the preferred items compared to the non-preferred items, you see that the ripple, this is triggered on the ripple event, is engaging in a link or co-activation with the representation in the cortex that are associated with these specific preferred items. So to summarize this part, what we found with this free recall paradigm, using language as a tool, we found that first it is regulated. Free behavior is regulated through general excitability shifts, baseline shifts in the activity of the neurons, and it is the events of recall in the absence of a stimulus are initiated through highly synchronized hippocampal population activity. Those bursts are probably initiating the emergence of an item in the mind of the observer. The next aspect I would like to discuss is social scale. It was mentioned already that large-scale social interactions are the foundation of human culture and human technology from ancient times, but in recent years we see a huge explosion of social scaling or social interaction mediated through the social media. And here I want to focus on a very narrow aspect of this explosion that is related to vision, and this is the formation of huge databases that contain labeled images of different categories. For example, in the case of faces, just to illustrate, Google now has a database of 200 million images of faces that are labeled by 8 million identities. So we have a huge set of images where we know each image face, what it is, who is the person that is part of this image. And this, of course, led to me, one of the most exciting developments in computer science, and this is the development of deep convolutional networks. I will not deal with details here because this will be discussed in later talks probably, but just to give you a stripped-down description of deep convolutional networks, the astonishing thing for me as a brain scientist, vision scientist, is that they now perform at the level of human performance in task of recognition. These actually, if you think about it, they are built out similarly like a stripped-down version of the human visual system. They have a hierarchical structure with layers starting with very early, the different neurons, artificial neurons in the layers start receiving an input, and it turns out that they have a very low level filters at the early layers, and then as you move up into the layers higher in the layer hierarchy, the neurons response or filters become, through convergence and various nonlinear filtering, they become more and more complex until at the end you have units that are actually point to you the identity of the image. In the case of object categories, will be the category of image. In case of faces, it will be who is this person, regardless of the images that are presented. And why were we were becoming interested in these networks? And this is work done with Shani Grossman. We encountered a very, very robust and uniform effect among the phase selective recordings that we were doing. And I want to illustrate this effect by showing you recordings from 2 electrodes that were done in the fusiform gyrus in a phase selective area. And these are recordings, responses to images of different kind of categories of object, different faces, different object categories. And first you see that this recording that I'm showing you showed a beautiful response to faces, but almost nothing to other categories. A classic phase selective response. But if you break down the response to the faces, to the individual images, we call them exemplars, you see that not all exemplars are activating this electrode at the same level. Woody Allen, I don't know if you see it here, but this Woody Allen activated much stronger this electrode than Barack Obama, for example. Okay? So this electrode showed, despite the fact that it is phase selective, a higher activation to one exemplar compared to others. Now you might say, okay, this patient likes Woody Allen more than Barack Obama, therefore all these visual face areas are actually activated more to Barack Obama. But if you look at the neighboring electrode, just a few millimeters away, you see again selectivity, but this time Barack Obama is the winner. He's the strongest response, and Queen Elizabeth is actually the weakest. So you see that there is a changing profile. We call it exemplar selectivity among these electrons. And this is a very robust phenomenon. We find it across all phase-selective electrodes. The ratio is typically twice the top exemplar compared to the bottom exemplar. And the question is, why do we have it? What does it mean? And the problem is we don't have a clue. This is the embarrassing answer. We don't know why you have this selectivity. And I want to contrast this problem because I think it's a general problem of finding tunings in high-order areas in the brain. When you are dealing with tuning in early areas, for example, area V1, it's clear to think about the function. It makes sense that these lines are actually functioning as edge detection. You can even build the wiring diagram that leads to the Nobel Prize for Hubel and Weizmann and also to the formation of this receptive field. But if you look at the high-order areas and you find some tuning to exemplar, what is this function? And the real worry is maybe it does not have any function. Maybe it's just some kind of an artifact, anatomical artifact. These are very reliable selectivities, but maybe they don't mean anything to the function of the system. So how can you get a handle on it? And I think Elon Vadia yesterday said it very nicely. The problem is that we don't have a model in which to fit those data and see how they function. That's where we became interested in the deep convolutional network, and we were wondering whether this could serve for us the sort of a toy model that will explain the exemplar selectivity that we find in the cortex. And how, you might say, but how can you even find any relationship between brain activity and artificial neural network, which is, after all, a software program? Here, there is an idea that was originated actually by Shimon Edelman and Kalanit Grill-Spector several years ago, and later on developed nicely by Nikos Grigas quartet. And the idea is that you should not look, when you're looking at neuronal responses in high-order representation, you should not look at individual electrodes. What's interesting is the pattern of activity to a particular image. So it's not what specific electrode is doing, but what is the pattern of response that you get for a particular image. And furthermore, what will tell you about the functionality, what about an area is doing, is how different patterns or different images relate to each other in terms of this pattern. So just to explain what I mean by that, if you compare this image to this image and look at the profile of response across electrodes that we have, this is assembling for many patients, you see that this profile and this profile for the same electrode is very, very different. We call this long distance separation between the two images. They are activating very different patterns. If you compare it to these two images, you see that here the pattern is very similar, so we call this short distance between the two images. And using this kind of logic, you can actually build a distant matrix between all the images that we show to the patient, similar to distant matrix that you have between towns in Israel, for example, where you can actually judge what is the distance, how similar are the patterns activated by the different pairs of images. The beautiful thing about this approach is that you can do exactly the same thing on the artificial neural network. You can take the artificial neurons, feed the network with the different pictures that the patient saw, and ask to what extent two different images that are fed to the network activate similar pattern. In that case, we'll say that they have short distance, or different patterns in the artificial network, then we'll say they have long distance, and we can construct the same distance matrix in the artificial networks to the one that we constructed in the human. And the beauty of it is that now you can go and compare the distant matrix that reflects the functionality of the visual areas in the patients to the artificial networks, and even more so, you can do it separately for each layer, so you can know which of the layers in this hierarchy of artificial layers fits best the functionality of the neurons that you found in the visual cortex. And this is what Shani did. And what she found is a very striking correlation between the brain activity recorded in the human patients and artificial neural network called VGG faces that were trained specifically on face images. And you can see here the scatter plots for these two highly significant correlations. These are scatter plots of pairs of images. What are the distances in the artificial network and the biological network? And you see very nice correlation, and you learn from it three important things. First of all, the selectivity that we found in the cortex was not artifact. It is replicated in the artificial network. Secondly, we have here a very interesting, you might call it convergent evolution between a computer and the human brain. You have here two systems, one biological and one artificial, that were trained to perform the same operation, and somehow the two systems arrived at a similar solution, at least in terms of reflection of the neuronal distances. And the third important message is that the correlation is not at the low level. In other words, it's not driven by this low-level artifact, and it's not at the highest level. It's not identity, but it's related to something in between, probably to the appearance of the faces. And this was not an incidental result. It was replicated on different data sets, and it was always higher for the face-trained networks compared to networks that just had the structure but had random weights. And finally, once you have a toy model like that, you can start doing endless manipulation. For example, you can ask, maybe the effect that we found was not because of the faces, but because of the different backgrounds that you see here. So you can now remove the backgrounds, generate this set of data, feed it into the artificial networks, and ask, will we maintain the same correlation to the brain? And you see the same exact correlation, ruling out the possibility that it's the background. And now you can manipulate many, many other parameters. So to summarize this part, the study, by comparing to deep convolutional network, we learned something about exemplar selectivity in human cortical face representation. First of all, we find this interesting convergent evolution. We find similar representational principles in the network and in the human brain. And the correlation actually tells us something about the function of this processing. It suggests that these representations are involved with the pictorial, with the appearance of the images, and not with the identity, because we did not find the correlation to the identity detecting units. Finally, I want to conclude with dealing with common sense. Now, I'll define common sense as information that is agreed by everybody. And the question that we want to deal here is with the issue, what can the human brain, I mean, visual perception is obviously not only exemplar selectivity, and obviously it's doing much more than deep artificial networks can do. And actually, I think it's an interesting topic of study now in the visual domain to find out in what way the visual perception is different from the performance of artificial networks. One of the things that we humans can do and these networks cannot do is to have bistable vision. If you look carefully at these images, you see that you can see the bouquet of flowers very, very nicely, but you might also see the faces that are hidden here. And once you see them, you can easily translate between seeing the faces or seeing the flowers. And the critical point to realize here is that nothing is changing in terms of the physical activation of the eyes. It's all happening in your mind as you're clicking back and forth between the two inner perceptions. And the question is, what is the neuronal mechanism that is doing this clicking or activating content, for example, that you see faces instead of flowers, without any change in the input? And we previously have suggested by several studies that there is a phenomenon, dynamic, selective, for example, when you see faces to the face selective areas that we call ignition, that was an amplification of the signal and persistence of the signal for several hundred milliseconds every time you saw a face instead of seeing something else. We did it with backward masking. And our suggestion was that this is because of recurrent activity local recurrent activity within the phase selective area. But the main criticism that we got is, and it's a fundamental criticism, is because you must, as I said, in order to know what the patient is doing, you must ask them, did you see a face, or did you see a flowers, or did you see noise? And the argument is, maybe all this ignition that you see has nothing to do with the perceptual aspect. It has to do with the fact that the patient is telling you that now I see this and not. In other words, it's related to the reporting about the percept and not the percept itself. And this is not a trivial methodological issue. This is a fundamental problem that has to do with all studies of subjective experience. We know that these experiences are private. We are the only one that actually know about them. So the only way we can study them experimentally is by asking the patient to tell us what's happening. But this also causes a confound, the possibility that telling is changing the activity. So we were searching with Tal Golan for some kind of situation that does not require a report. And we call this situation common sense illusions. And the idea is that there are certain illusions where you have a dissociation between what you see and the stimulus, the physical stimulus, that are so powerful and so prevalent and happen to everybody that there is a common sense agreement that we don't need to ask for reporting. And what do I mean by that? In the last hour that you will listen to me and the other speakers, without noticing that, every 5 minutes, your visual input was completely interrupted, okay, momentarily. And you are, until this moment, were not aware at all that that happened to you. You had a continuous experience of the visual world. And this is because you are spontaneously blinking about every 5 seconds. And these spontaneous blinks actually constitute quite a long time of eye closure throughout the daytime. And despite the fact that they are causing an abrupt and complete interruption of the visual information flowing into your eyes, you are completely oblivious to it. And I don't think I need to argue with anybody with you who would say, no, I see blinks, I see darkening every time I blink my eyes. It is a common sense agreement that we don't see it. But if I simulate those blinks, in other words, I create a situation of the same blockage that you have during blinking, then you see them very, very clearly. For example, this is a simulation that Tal did. He simply blocked the image every time the eyelids close, and you can, I think, agree that you see a very striking interruption. So this leads us to allow us to do experiments trying to see what is the difference in the brain when you are equating the interruption, but sometimes you don't see it because you spontaneously blink, and sometimes you saw it because I introduced it artificially. I will call this artificial introduction as gaps. So we simply compare gaps that the patients see with spontaneous blinks that we measure with eye trackers while the patients are watching images, and we ask where in the brain there will be a reflection of the perceptual content, and where in the brain there will be a reflection of the optical content. So let's first look at area V1. This is an example electrode. You see beautiful response. And when we are doing the gap, in other words, we are interrupting the visual stimulus in such a way that the patient sees the interruption, we see that V1 is activated very robustly, both to the disappearance and the reappearance of the image. But this is not surprising. We interrupted the visual input, and we interrupted psychologically. In other words, we saw the interruption. The interesting thing is the question, what happened during the spontaneous blinks that are completely invisible. You see that V1 is as robustly activated as when the images are visible. It appears from this data that area V1 does not actually participate in perceptual experience. It's of course the channel through which information is going, but we are not reading the activity in V1 because you see here a huge amount of activity in V1 that we are insensitive to. If you now look in high order areas, for example, phase selective region, you see during the spontaneous blink a complete shut-off of response. In other words, these areas go with the lack of interruption that is happening during spontaneous blinks. I remind you, you don't see the spontaneous blink. But if you look during the artificial blink, the gaps, then you see a robust activation that is related to the fact that we see the interruption. So high-order areas are reflecting with this on or non-ignition phenomena the perceptual state of the observer. So in the absence of report, what we find is that early visual cortex shows massive activation that remains completely invisible to the individual, but high-order areas show an on or non-ignition effect that completely shuts off if you don't see the stimulus. And just to summarize with a bit of speculation, and this is indeed a highly speculative summary, I would like to suggest that maybe the selectivity of high-order cortical neurons is generated by subconscious feed-forward networks, just like deep neuronal networks are doing, and the perceptual emergence, the fact that a percept emerges in your mind, is due to recurrent activity that forms the dynamics of an ignition that binds together these selectivities to give you an ensemble that represents the gestalt properties of these neurons. And the free behavior is regulated by changing the temperature of the tissue, so to speak, and is driven by this really enormous burst of synchronized activity called ripples in the human hippocampus. Thank you. I would like to give credit to the individuals that participated. Itzhak Norman did the study on the free behavior. Shani did the study on the relation to DNN. Tal Golan did the study on blinks. Professor Mehta is the neurosurgeon with whom we collaborate on the patients. And Guy Gaziv, I didn't mention, is helping us in the deep neural networks. He's from the lab, computer vision lab of Michal Irami at the Weizmann Institute. And I want to mention specifically Michal Harel, who was responsible for the anatomy, is the general mother of the entire lab. Thank you.
Thanks very much. Wonderful talk. While we are switching speakers, I think we have time for 1 or 2 questions.
Yes.
Thank you very much. I was wondering if it's only the higher level that recognizes or that doesn't, or let's say differently, recognizes the difference between a blink and an artificial one. Where does it get the information from?
The information of
the difference between the artificial and the...
I think there is a gating between the early visual areas and high-order areas. You saw it very beautifully. And we think that it came from premotor areas that are actually sending commands to blink and have what's called an efferent copy pathway that is sending information not only to the motor muscles that blink, but in parallel to the visual system to cause the gating. We're now studying various kinds of predictions to see what is the mechanism that actually cause the differentiation between the blinks, which come with knowledge from inside that you are going to blink, and the gaps that you don't know about, they come as surprise. So there's an issue here of prediction and surprise that is very interesting.
On the same line, Rafi, here, Elon is here. On the same line, when you told us that we didn't notice the blinks, on that moment on, I had dark moments for the rest of the lecture.
Hopefully not for the rest of your life. Well, that's a very good point, because if I ask you to voluntarily blink, you will see the darkening. We think there is an issue of attention here that I didn't have to come into. Part of the fact that you don't see the blinks is because you don't attend to them. The moment you attend to them, you start feeling them. By the way, I started noticing when people that I talk to blink, and there's a whole interesting message that goes on there as well. So it's an interesting topic to walk around and think about.
Thanks much again.